Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this lecture, I will be going over pyrimidine synthesis and also pyrimidine degradation. Let's get into the details. Now, unlike purine biosynthesis, purine ring biosynthesis, where a PRPP, that is phosphoribosyl pyrophosphate, is the one that is necessary initially, over which each and individual atoms for purine nucleotide is built. Okay. Whereas here in pyrimidine nucleotide biosynthesis, the pyrimidine ring is synthesized first and then PRPP is added later. And that's the major difference between purine nucleotide biosynthesis and pyrimidine nucleotide biosynthesis. That means pyrimidine base is synthesized first here and later PRPP is added. Now, very first step it involves in pyrimidine biosynthesis is the formation of carbamoyl phosphate, which is done by carbamoyl phosphate synthetase 2 enzyme. And this carbamoyl phosphate synthetase 2 enzyme is the most regulated enzyme in pyrimidine biosynthesis, which is there in the cytoplasm. It's a cytoplasmic enzyme. And there is a similar enzyme called carbamoyl phosphate synthetase 1. It is there in the mitochondria and participating in the urea cycle. So, CPS-I is there in the urea cycle, whereas CPS-II enzyme, it is there in the pyrimidine biosynthesis. Both are most regulated enzymes for each of these metabolic pathways. Let's look into little more details on CPS-I and CPS-II. So, CPS-I present in urea cycle, CPS-II, it is present in pyrimidine biosynthesis pathway. And the source of nitrogen for CPS-I, it is ammonium ion directly, whereas source of nitrogen for CPS-II is glutamine. Location is CPS-I is mitochondria in the urea cycle, whereas CPS-II, it is present in cytosol for pyrimidine biosynthesis. And the activator of CPS-I is N-acetyl glutamate, NAG, and the activator of CPS-II is PRPP. Remember, PRPP is going to stimulate both purine and pyrimidine biosynthesis. And inhibitor of CPS-II is UTP, and there is no such inhibitor on CPS-I enzyme. So these are the major differences between CPS-I and CPS-II enzyme. Now let's get into de novo synthesis of pyrimidine nucleotide biosynthesis. So it all starts from condensation of carbon dioxide with the glutamine and ATP to make carbamoyl phosphate, done by carbamoyl phosphate synthetase 2 enzyme. It is the most regulated enzyme in pyrimidine biosynthesis. Remember that. So once you get that nitrogen coming from glutamine there, so then the aspartate comes in. Almost entire molecule of aspartate is added on to carbamoyl phosphate, done by aspartate transcarbamylase enzyme. It is the intermediate step in pyrimidine biosynthesis. So it is a rate determining enzyme for pyrimidine biosynthesis. It is aspartate transcarbamylase. Most regulated enzyme is carbamoyl phosphate synthetase 2, whereas the most rate determining enzyme or committed step is catalyzed by aspartate transcarbamylase. So we are going to get carbamoyl aspartate. Now this carbamoyl aspartate with the help of dihydroorotate enzyme, there will be a ring closure and you get dihydroorotate. And this dihydroorotate undergoes oxidation process to make orotate. So that's how we get orotate. That means reaction number 1, reaction number 2 and reaction number 3. So it is carbamoyl phosphate synthetase 2, aspartate transcarbamylase and dihydroorotate enzyme is they are all present in one big enzyme complex called CAD trifunctional enzyme. CAD trifunctional enzyme. So CAD trifunctional enzyme basically has 3 enzymatic activity. That is carbamoyl phosphate synthetase 2, aspartate transcarbamylase and dihydroorotate. All of them are present in one single functional enzyme complex called CAD trifunctional enzyme. Okay, once you get orotate here, so done by dihydroorotate dehydrogenase. Remember, this is inhibited by a drug called leflunomide, which I am going to show you in the later slides. Once you get orotate, now the orotate is it's a pyrimidine ring here already. Now you get PRPP, phosphoribosyl pyrophosphate, which is brought in by orotate phosphoribosyl transferase enzyme, OPRT. orotate phosphorybossyl transferase enzyme transfers that PRPP and it makes orotidilic acid monophosphate OMP. That's a pyrimidine nucleotide here. Pyrimidine base orotate is converted into pyrimidine nucleotide now by transferring sugar and phosphate, ribose and phosphate. Now OMP or orotidilate monophosphate is undergoing a decarboxylation to become, that's done by orotidilic acid decarboxylase enzyme to become UMP, uridylate monophosphate, UMP. Now the UMP is the first true pyrimidine, that is UMP, uridine monophosphate. Now the uridine monophosphate will be converted to uridine diphosphate by kinase enzyme, and that uridine diphosphate converted to uridine triphosphate by another kinase enzyme. So that's how we got UTP for RNA synthesis now. Okay, so we have two enzymes here in the formation of UMP, uridine monophosphate, that is conversion of orotate into orotidilate monophosphate by orotate phosphorybossyl transferase OPRT, and then OMP is converted into UMP by orotidilic acid decarboxylase or orotate decarboxylase enzyme. So both OPRT and orotidilic acid decarboxylase, they are present in a single enzyme complex that is called as UMP synthase. So UMP synthase, it has two enzymatic subunit. One is orotate phosphorybossyl transferase, other is orotidilic acid decarboxylase. Remember that. So if there is any deficiency or defect in UMP synthase, that leads to elevation of orotate that appears in the blood and urine, leading to orotic aciduria. So UMP synthase defect may lead to orotic aciduria, and UMP synthase is combination of two enzymes. So OPRT can be defective or orotidilic acid decarboxylase enzyme can be defective. We are going to see that later. Okay, so we got UTP now to make RNA. So what about other pyrimidine nucleotides? So UTP can undergo a reaction where ammonium nitrogen coming from glutamine is taken in, done by CTP synthase to make CTP. So we got CTP there, which can go into RNA formation. So UTP and CTP can go into RNA synthesis now. What about DNA? So UDP, uridine monophosphate, can be catalyzed by ribonucleotide reductase enzyme, RR enzyme, which uses NADPH plus to convert UDP into deoxyuridine monophosphate. Basically the ribose sugar is converted to deoxyribose sugar. That's how UDP is converted to deoxy-UDP, and the deoxy-UDP undergoes phosphorylation reaction becoming a deoxyuridine monophosphate, DUMP. So DUMP is converted to DTMP, that is done by thymidylate synthase enzyme, and for that the coenzyme is N5N10 methylene tetrahydrofolate, and this N5N10 methylene tetrahydrofolate in turn will be coming out of the reaction as dihydrofolate. Now this dihydrofolate will be converted into tetrahydrofolate first, done by DHFR, that is dihydrofolate reductase, and tetrahydrofolate further converted into N5N10 methylene tetrahydrofolate. So there can be drugs which can inhibit these certain enzymes in this pathway, that is 5-fluorouracil, 5-FU will inhibit thymidylate synthase enzyme, thereby it decreases the conversion of DUMP into DTMP. That's a direct action of 5-fluorouracil on thymidylate synthase. Or methotrexate, it can inhibit dihydrofolate reductase, DHFR enzyme, thereby decreases the conversion of dihydrofolate into tetrahydrofolate, thereby it decreases the availability of N5N10 methylene tetrahydrofolate, and that indirectly will affect functioning of thymidylate synthase. So methotrexate has a direct effect on DHFR and it has an indirect effect on thymidylate synthase enzyme, whereas 5-fluorouracil, it has a direct effect on thymidylate synthase enzyme. So this is how we make DTMP. Once you get DTMP, you can convert that into DTDP, and that DTDP can be converted into DTTP, and that can go into DNA formation. What about this cytidine, deoxycytidine triphosphate? So CTP here converted to CDP, and CDP into DCDP, and done by ribonucleotide reductase, DCDP into DCTP, and that DCTP can go into DNA synthesis, cytidine triphosphate. So thymidine triphosphate, cytidine triphosphate going into DNA formation. So like this, pyrimidines are synthesized for both RNA and DNA synthesis. So all the steps that I have explained so far, I have written here. Carbamoyl phosphate synthetase 2 is the regulated enzyme, and formation of carbamoyl aspartate is the committed step. That's the second step. Entire aspartate molecule is used in the pyrimidine ring formation. Leflunomide inhibits dihydroorotate dehydrogenase enzyme. UMP synthase is a bifunctional enzyme that makes UMP, and a defect in UMP synthase will lead to orotic aciduria. Remember that. Now, UMP is converted to UDP by UMP kinase, and then UTP is made. And CTP synthase adds amino group from glutamine side chain to become, so basically to convert UTP into CTP, and UDP can undergo ribonucleotide reductase reaction to make DUDP and then into DUMP, and DUMP can be converted to DTMP by thymidylate synthase enzyme, and 5-fluorouracil can inhibit thymidylate synthase enzyme. Similarly, methotrexate by inhibiting dihydrofolate reductase enzyme, it is going to decrease the availability of N5-N10 methylene tetrahydrofolate, thereby indirectly affecting the conversion of DUMP into DTMP formation. Coming to regulation of de novo pyrimidine biosynthesis. Now, as I told you already, CPS-II is the most regulated enzyme in the pyrimidine biosynthesis pathway. So that means it is positively modulated by PRPP. PRPP, it is allosterically activated by PRPP and ATP. Both of them are positive modulators on CPS-II enzyme. Okay, so PRPP is positive and ATP also act as positively on CPS-II enzyme, thereby increases the formation of carbamoyl phosphate, and that carbamoyl phosphate going into carbamoyl aspartate formation, and then all the way into OMP, UMP, UDP, CTP and all that. Okay, so UMP, it has a competitive feedback effect on orotidylic acid decarboxylase enzyme, and whereas CTP it is going to inhibit CTP synthase enzyme, and it basically balances between synthesis of uridine and cytidine nucleotides. And also remember, CPS-II helps in decreasing ammonia toxicity, because in whenever there is a defect in urea cycle, so whenever urea cycle is kind of saturated, so the carbamoyl phosphate from the mitochondria, very first reaction in the urea cycle, the product is carbamoyl phosphate. It is coming out of the mitochondrial matrix into the cytoplasm, and in the cytoplasm carbamoyl phosphate is placed into pyrimidine biosynthesis, and it is neutralized. Basically, there will be orotidylic acid or orotate which will be elevated, and that will go out of our body as orotidylic acid, which is a less toxic molecule than ammonia. So that's how CPS-II, it helps in decreasing ammonia toxicity. Remember that. Alright, so coming to degradation of purine bases. So the purine bases, that is cytosine, uracil and thymine. All these three purine bases, ultimately they can be converted into water soluble end products, that is you can get carbon dioxide, ammonium ion, beta alanine, beta amino isobutyrate molecule. So they all will undergo degradation in a sequential way to produce all water soluble end products. So generally pyrimidine degradation products do not pose any serious health effects, unlike purine degradation product. Remember that. So the overproduction of PRPP can lead to increased pyrimidine, that leads to elevation of beta alanine. That's the only thing that we can see. Whereas the overproduction of PRPP in terms of purine nucleotide biosynthesis will lead to elevation of uric acid, which can lead to uric acid stone and even renal failure. But that kind of effects, serious effects, we don't see with the pyrimidine degradation pathway. Coming to one of the congenital disorder associated with purine nucleotide biosynthesis, and that is orotic aciduria. So orotic aciduria is because of autosomal recessive mutation in UMP synthase gene, which is located on chromosome number 3q13. Now, what is this UMP synthase gene doing for? So, UMP synthase gene is coding for an enzyme UMP synthase, which is a bifunctional enzyme that contains OPRT and also orotidylic acid decarboxylase activity. So, if there is a deficiency of both the enzyme, that entire UMP synthase is down, that leads to type 1 orotic aciduria. Or if it is only orotidylic acid decarboxylase component of UMP synthase is defective, that leads to type 2 orotic aciduria. So, whether it is type 1 or type 2 orotic aciduria, patients with orotic aciduria will manifest with megaloblastic anemia and excess orotic acid being elevated in the urine, leading to orotate crystallization. So, orotic acid crystals can be seen in the urine. So, that is orotic acid crystalluria. So, why do you see megaloblastic anemia and orotic aciduria? It's because when pyrimidine synthesis is decreased, so when the lack of purines and pyrimidines are there, so that means there will be decrease in the formation of DNA and RNA. So, nuclear size becomes small, cytoplasm is compensated. So, that leads to nucleocytoplasmic disproportion and red blood cells will become big, leading to megaloblasts. So, maturation of red blood cell will be affected. Along with the megaloblastic anemia, so and orotic crystalluria, patients with orotic aciduria will also manifest with mental retardation. So, the phenotype features of orotic aciduria are megaloblastic anemia that is not responsive to vitamin B12, folic acid or iron or pyrimidine, iron or pyridoxine supplementation. Remember that. So, it is resistant to supplementation to cobalamin, folic acid, iron and pyridoxine. So, that means the megaloblastic anemia is other than vitamin B12 and folic acid deficiency, and that is because of UMP synthase enzyme being defective here. So, how do you treat congenital orotic aciduria? It is, you can treat it by using uridine as the uridine supplementation. Basically, the uridine nucleoside can be given, and once you give uridine nucleoside, so uridine nucleoside inside, it can be used to make UMP, and once you make UMP, you can make CTP and DUMP, which can be converted into DTMP. So, all pyrimidine nucleotides can be synthesized once uridine is provided, and that will lead to reversal of signs and symptoms seen in orotic aciduria. So, this is all about pyrimidine metabolism, both pyrimidine synthesis and pyrimidine degradation, including many drugs that are involved in the pyrimidine metabolism, which are affected and leading to certain treatments. That's about it. Thank you.